Hello everyone and welcome to today's webinar, Quality Control Advancements in Therapeutic Genome Editing and Biomanufacturing. I'm Mark Lynch of Genomic Vision and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Genomic Vision. To learn more, visit genomicvision.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Imen Mestiri, Scientific Account Manager at Genomic Vision. Imen, you may begin your presentation.
Thank you, Mark, for the introduction. My name is Imen Mestiri. I'm a Scientific Account Manager at Genomic Vision. Okay, welcome to our summer webinar series where we are on molecular combing and its applications. So during the first session, we presented molecular combing principles and how it's transforming the genetic research through the analysis of single genome molecule. During the second session, you learned how molecular combing can simplify complex genetic diseases by quantifying structural and copy number variants. And during the third session, we presented how molecular combing improved understanding of the replication kinetics in oncology field. And I'm happy to present today our last webinar session on quality control advancement in therapeutic genome editing and biomanufacturing. Molecular combing is a single molecule-based technology, and we use it for many applications. For example, in human genetics, in cancer research, in drug development, and also in genome engineering and novel therapies. And this will be the focus of my talk today. So I will present Genomic Vision-based quality control assay for therapeutic genome editing and animal model generation and biomanufacturing. And I will start my talk with the application of molecular combing to therapeutic genome editing. Genome editing and the development of the CRISPR-Cas system has really revolutionized the way we really see science. In basic research, it facilitates the development of cellular and animal models for functional studies. In the therapeutic field, it accelerated the development of ex vivo and in vivo therapies for diseases with unmet needs. It is now also being used to engineer better cell factories for the production of the biologics, like, for example, recombinant proteins, monoclonal antibodies, or vaccines. And last but not least, it accelerated the development of plants and animals with the desired agronomic traits. In the therapeutic field, so there is several clinical studies that using gene editing tools and also the CRISPR-Cas system that have already started. And the first results from the clinical trials are very promising, but there is still some safety issues that need to be addressed with using these gene editing tools. Mainly the p53-mediated response to double-strand breaks that can lead to cancer formation, off-target effects, and also on-target rearrangement that are caused by the DNA repair machinery, and that can be quite complex. And lastly, translocation that can be caused by potential off-targets or multiplexing strategies. So the regulatory agencies recommend to identify and characterize off-targets and also chromosomal translocation, and to evaluate the profile of insertion and deletions and all type of mutation generated at the targeted locus. So it's really important to accurately characterize and quantify gene editing events in order to mitigate the risk-benefit balance and bring this tool safely to the clinic. Genomic Vision, we provide a technology that combines direct visualization, single molecule analysis, and a panoramic view of the targeted regions. So molecular combing can visualize and quantify mid-SARS variants that are between 2 and 10 kb in size, quantify large variants and translocations that can be more than 10 kb in size. It can also visualize and quantify on-target rearrangement that can be quite complex, like, for example, inversions, duplications, that can happen a couple of kb away from the target site, and also exogenous DNA integrations. So as an example, if you are using AAV to deliver the CRISPR-Cas9 components or the therapeutic protein, we can monitor for potential integration of this AAV into the induced double-strand breaks. So off-target detection is partially possible with molecular combing, and it requires a prior nomination of this off-target by another technology. So the rod-blood digital PCR, short and long-read sequencing can also fish are powerful technologies and can be used to assess gene-edited outcomes. But none of these technologies combines the visual and quantitative aspects that is really unique to molecular combing. So molecular combing can quantify the efficiency and fidelity of gene editing-based therapies. And the principle is quite simple. So if, for example, you are aiming to induce a targeted deletion using CRISPR-Cas9, we will design a set of probes that are specific to your target locus. We will then visualize the obtained signals and classify them in different categories, so in either edited molecules, or rearranged molecules. And the rearranged molecules can have a variety of rearrangement at the targeted locus. So we will then quantify each signals per each molecules per category of signals. So the efficiency is defined by the number of edited molecules divided by the total number of molecules, and the fidelity is defined by the number of rearranged molecules and the total number of molecules. And this is how we will we assess the gene edited outcomes. And I will give now two case studies to illustrate what I'm just saying. So in the first study, we used a molecular combing to validate a 2 kb targeted deletion induced by the CRISPR-Cas9 system. So primary fibroblasts that are derived from patients harboring a pathogenic mutation between the exon 15 and the exon 16 were electroporated with a ribonucleoprotein complex to induce a 2 kb deletion at the target site. So we designed a set of probes that are composed of probes of different size, color, and spacing, and that create a unique barcode that we call GMC for Genomics Morse code that is really specific to the target locus. And the scheme of this GMC is really depicted here at the bottom of the slide. And so here are some results. And so as a reminder, the GMC barcode is really presented at the top, and it covers a region of 40 kb in size. So the targeted deletion is labeled in green, and it's really the fragment number named C. So in the first panel, this shows the unedited events. So as you can see here, this is examples of signals that we can obtain with our fiber vision scanner. And the C fragment in green here is really still present, as you can see very clearly. And the number of these events is around 80.7% frequency. So in the second panel, we show editing events. And you can clearly see here that the green fragment is absent. And the efficiency of our editing is around 17.7%. So importantly, in the same sample, we also observed on-target unintended rearrangements that are depicted in the third panel. So, and these events are caused by sister chromatin fusions and happens at 1.6% frequency. And this is quite high, and it's really difficult to identify this type of rearrangement with another technology. And what is really nice with molecular combing is that you can see clearly what is happening, and this gives you also a hint of what type of mechanism and what type of mechanism led to these rearrangements. So we scored more than 1,000 signals per sample to provide a robust safety assessment to our clients. As a summary, molecular combing is a powerful tool to visualize edited signals and quantify their frequency, to visualize and quantify on-target rearrangements, especially unbalanced translocation, and also to provide a mechanistic understanding of the identified rearrangements. I will present now the second case study where we monitored CRISPR-Cas9 gene editing event at the BRCA1 locus. So HEC293 cells were electroporated with an RNP complex to generate a 6.5 kb deletion. In this case, we also compared 3 guide RNA pairs, and the position of the left and right guide RNAs is depicted at the bottom of the slide. So we then detected and quantified intended gene editing events. So in the image presented here, on the top, you will have signals from a wild type allele. So the B sequence that we intended to delete is still present. And at the bottom, you will have an example of the edited allele. As you can see, the B fragment is deleted, indicated that the editant was efficient. And we quantified the efficiency of this editing that ranges between 6.5 to 11%, depending on the guide RNA pairs used. Importantly, and like in the first case that I showed, we also observed on-target rearrangements. And so the first, previously, the wild type allele is depicted at the top of the image, and the rearranged alleles at the bottom. And as you can see here, the rearrangement that we observed is quite complex. So the BA fragment has been deleted, so there's a deletion, and also an inverted duplication at the same time. So we quantified the frequency of these events that ranged between 2.6 to 4%. Depending on the guide RNA pairs. We also observed some rearrangement in the control that we used, and this is also linked to the fact that the BRCA1 locus is a fragile site that is subject to rearrangement in untreated conditions. So it really also pinpoints the importance of control in the context of gene editing in order really to differentiate the editing effects from the naturally occurring and basal structural variants. So we then sought to compare our deletion frequencies with an alternative to the technologies, and we then used a droplet digital PCR. So the deletion frequencies calculated by droplet digital PCR is presented in the graph here, and the range of deletion frequencies is very similar to what we observed with combing, validating our results. So droplet digital PCR allows for an absolute quantification of targeted deletion, but it's really blind to other rearrangements like the complex one that I just presented or the cisted chromatid fusion that I presented in the first case. As a summary, molecular combing is unique in the sense that it enables both visualization and quantification of intended and unintended editing outcomes in a single experiment. It can detect and quantify large targeted deletions without amplification of capture bias. It quantifies the efficiency and the fidelity of gene editing events. Unintended on-target rearrangements happen at a high frequency, and sister chromatid fusion are the most common rearrangements observed. So whether these rearrangements are persist or are counter-selected in further divisions is still an open question and needs further investigations. So I will now present the application of molecular combing to transgenic animal models. So transgenic animal models are very important for basic research and also for preclinical studies. And it's really key to accurately validate the engineered animals. And why? And here I'm going to quote one of my clients, because the production of transgenic animal models by traditional pronuclear injection results in a random integration of single and often multiple copies of transgenes that can be arranged in concatenators. And this can have a profound effect on transgene expression. It is though critical to deeply characterize the transgene integration site and also the pattern of integration to conclude that the desired phenotype is associated with the transgene. So we used molecular combing to characterize the transgene integration pattern. So we designed a 3-color barcode that is specific to our transgene, and we found that the transgene integrated as a 600 kb concatenator of 40 copies. So an example of signals from this concatenator is presented here, and the number of copies are annotated on the top. So from the copy number 1 to the copy number 10, we found that the transgenes are arranged in a head-to-tail orientation. The copy number 9 was found to be rearranged, so it's a truncated copy. And as you can see in the enlargement of the signal, the blue fragment that is encoding the gene of interest is deleted. So from copy number 10 to copy number 40, there is a change in orientations, and all the copies are oriented in a tail-to-head orientation. So molecular combing really allows for fine characterization of transgene integration patterns, and this deep characterization of transgenes is really key in the field of gene and cell therapy, and for animal model generations, like the generation of humanized mouse models, but also it's a regulatory requirement in the bioproduction field. And I will present some examples in my talk now. So a population of cells that are usually clonally derived is used to produce a biologic that can be a recombinant protein, a vaccine, or monoclonal antibodies. And it's really important to characterize the transgenes during the cell line development and banking in order to minimize potential risk and also to ensure consistent product quality and manufacturing stability. As to the current guidelines for transgene characterization, they recommend to document the coding sequence of the expression construct, the copy number of the transgene, its structural integrity, and also its integration pattern. Molecular combing-based quality control meet with these requirements. So our molecular combing-based quality control can be used throughout the cell line development pipeline. It can be used for screening and clone selection after cell engineering or single-cell cloning, and it can be used for genetic characterization and genetic stability testing during cell line expansion and banking. So our quality control assay can identify the vector identity, its integrity and copy number, and also co-integration events, like, for example, bacterial contamination, if this is something that is suspected, and last but not least, structural rearrangements that can happen in the transgene or also in the target locus. So we can both monitor random integrations and also targeted integration in the case if you are using the CRISPR-Cas9 system to introduce your transgene into safe harbor to have a consistent expression of this transgene. So we can, of course, estimate the number of copy numbers or provide an absolute quantification of these copies depending on the integration strategy that you are using. We'll provide the characterization of the integration pattern and also quantification of potential rearrangements that can happen at the targeted locus in case of the targeted transgene integration. So Genomic Vision is supporting several pharmaceutical companies, including Sanofi Genzyme, where we used our quality control assay to characterize their cell banking process. And we are also a member of the Niche Genome in a consortium that conveys different stakeholders in the gene editing fields. And the aim of this consortium is really to develop standards and methods that are needed to improve the confidence of using these genome editing tools. To conclude, I really hope that I convinced you today that through the analysis of hundreds of single DNA molecules, molecular combing represents a powerful analytical tool to generate high-quality cell lines and monitor their stability throughout the manufacturing process. And this is really a critical step in the development and production of any biological drug. So molecular combing can also be used to help developers of gene and cell therapies to monitor unintended biological consequences of genome engineering in order to refine their strategy and to bring safer therapies into the clinic. As a take-home message, molecular combing is a quantitative method. It allows single molecule counting of hybridization signals without amplification bias. It is a sensitive method, so we can go up to 1,500 equivalent genome per sample to reach a sensitivity of 0.25%, and this really allows to identify rare events. It is a visual method, and this is really key for molecular combing because it allows a long-range view of the targeted area. And this will help in identifying large and complex patterns, including concatenators, like I showed in the example during my presentation. And this type of complex structure are usually missed by next generation sequencing. And lastly, it's really orthogonal method to NGS and PCR-based assay. So our quality control assay is provided as a service, and we have different type of services that can be tailored to your needs. So the EasyComb and the EasyComb Plus, it's really start from the sample. You send us the sample, we take your samples, and we take care of the rest. So either we send you a full report for the EasyComb Plus, or we give you access to our software to analyze your data if needed, and this will be the EasyComb, just the EasyComb. For the EasyScan, so it's really for an access to our imaging system, and this is for if you want to set up the technology in-house. Thank you very much for your attention, and if you want to know more about our services and products, please contact us at sales@genomicvision.com. We would happily schedule a call to discuss your project and needs. So all our summer webinar series are now available on demand. And I thank you again for your attention, and I'm happy to take questions.
Thanks, Imen, for the informative presentation today. We'll now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. With that, let's get started. Imen, the first question that's coming in is about the services and how this actually works. So the question is, do you provide services? I saw in your presentation that we do, but how do those services actually work? Could you elaborate a little bit more on that, please? Imen, we're having some difficulty hearing you.
Okay, so I was on mute.
Okay, could you answer that question? Thanks.
Yes. Yeah, so we do provide services, and these services are tailored to your needs. So we have 4 types of services. So we have the full the end-to-end service solution. So you send us your samples, we do the molecular combing, the hybridization, the scanning, and the analysis of the data, and then we send you full report. So we also have another service where we stop at we do the experimental part and then the scanning, and we give you access to our software so that you can analyze the data yourself. So this is what we call EasyComb, and EasyComb Plus comes with the full analytical part and the report. Then we also have what we call an EasyScan, and so this is an access to our scanning system. And for this, if you are interested in setting up the technology in-house, you can purchase our fiber comb system, and we will accompany you and help you in getting combing up and running in your lab, and then we will give you access to the scanning services and the software itself. So this is how our services work.
Thanks, Imen. The next question that's coming through is, what is the sensitivity in general of molecular combing?
So with molecular combing, we can reach a sensitivity of 0.25%. So this is something that we do in a routine, and we can also go lower. We have some example where we could see some rearrangement at a very low frequency, and so this is also we can go below 0.25%.
Okay, thanks, Imen. Another question that's coming through, is it possible to identify inversions with the molecular combing technology?
Yeah, absolutely. So the principle would be very similar to what I showed for the deletions. So as long as we can design a barcode that will allow us to identify the inversion, so it's possible. It's just we can do it for inversion that are bigger than 3 kb. So lower than 3 kb, it will be challenging. But everything that will be bigger than 3 kb, it's totally possible.
Okay, thanks, Imen. Another question coming through, and I apologize, I do not know what AAV is, but let me read out the question anyway. Do you have any experience with AAV? Is the resolution sufficient to visualize paternal or paternal AAV integration at cut site?
Okay, so AAV is adeno-associated viruses. So we do not have experience with AAV itself, but as long as so the AAV usually is around, I think, between 4 and 4.7 kb. So it's really in our range of resolution. So we can design a set of probes that is specific to the AAV, and we can of course monitor if this AAV integrates at a particular locus. So it's really something that we can monitor. And we know that from some publication in the literature, AAV can integrate in double-strand break that are induced by the use of gene editing tools. So we can monitor the integration of AAV.
Okay, thanks, Imen. We're showing one more question here on the list, and that is how frequent these sister chromatid fusions events are.
Okay. From our experience, with the range that we found, it varies between 0.2 to 4%. So it's variable, and I think it also depends on the locus and maybe on the targeted locus and its position, if it's telomeric or centromeric. So this is so the frequency is variable. And it depends on the position. And this is also what was found by a group in Dana-Farber that published it also on chromotripsis, I guess, and that found that sister chromatid fusion also happened at a similar range.
Okay, thanks, Imen. Hopefully that answered the questions of the audience. We are not showing any further questions. So with that, I'd like to thank again Imen for your time today and your important research and presentation here. We'd also like to thank LabRoots for facilitating today's educational webcast. Before we go, I'd like to thank also the audience for joining us today and for their interesting questions. For any questions that are still pending, you can still submit those during the on-demand period, and they'll be addressed via the speaker via the contact information you provided at the time of registration. This webcast, along with the other 3 webcasts from Genomic Vision, are available on demand. LabRoots will alert you via email when it's available for replay, and we encourage that you share that email with your colleagues who may have missed today's live event. Thank you so much for all your time and participation in today's webinar, and for those that attended all 4 webinars, we thank you very much for that. Thank you and good day.